In this video, we're going to look at how PCR and RT-PCR, which stands for reverse transcriptase PCR, are used to detect infectious diseases, in this case, SARS-CoV-2. A few basic notes before moving on to looking at these steps individually in more detail in the lab. So, PCR or RT-PCR consists of three main steps. Obviously, the sample needs to be obtained from the patient using a swab that is mostly done through the nose or through the pharynx. There's another way, which is endotracheal aspiration, which is used less commonly but gives us a more specific and sensitive result. And then there is a pre-PCR step, which is done through a lysis buffer. We're going to look at this in more detail in a bit. And then there is RT-PCR machine, which is obviously the last step, and then the sample is placed there, giving us the results. Just a few more details about RT-PCR and PCR. And this looks like a crowded picture, but it's actually a very good representation of what happens. So we'll go through it. We've already looked at steps 1 and 2 in this picture, but we haven't really explored step 3, which is RNA extraction. So this step is necessary because PCR or RT-PCR really requires only the genomic material, so either RNA or DNA, of the virus or the bacteria. So in this case, what we're doing is extracting the RNA only of the SARS-CoV-2 virus, and we are lysing, destroying other materials such as protein or any other human cells that cannot be processed in the PCR machine. As mentioned previously, SARS-CoV-2 is an RNA virus, and PCR only works if there is DNA, so it can amplify DNA only. So a step that is additionally needed for this specific virus is reverse transcription, which means the conversion of RNA, viral RNA, into DNA, which can then be amplified by normal PCR processes. The result of this process is the production of complementary DNA, or cDNA, which would then be used as normal DNA in the PCR process. So now we're going to look at different simplified steps of PCR. As mentioned before, cDNA, which is also called the complementary DNA, which was formed from the process of reverse transcription, is needed for PCR. The first step is called denaturation. It causes the double helix of the cDNA to separate, exposing each nucleotide. The second step is called annealing. That's when DNA primers are added to very specific sequences of the viral DNA. These sequences have to be unique to that bacteria or virus that we are intending to study. And these genes are found by scientists. For SARS-CoV-2, there are usually 3 genes that are used in PCR. There is the RDRP gene, there is the E gene, and there's the N gene. In the lab that we're going to see, the RDRP and the N gene are being used in the PCR process. The consequence of using very unique and specific genes of the virus or the bacteria that we intend to study is that only these genes of the bacteria or virus that we intend to study are going to be amplified in the PCR process. So any other genetic material, such as that of the human host or any other bacteria or virus present there inside the swab sample, will not be amplified. And finally, after the annealing process, elongation occurs, where there is more nucleotides being added to the sequence. We will now look at all these processes that we have described earlier inside the lab. The samples obtained from the patients come in these containers with the patient's details on them. They're covered by 2 biohazard containers. Here, we see where extraction is done. It is primarily done by these 2 devices. For extraction to occur, we need to place samples from the patient's PCR into this container. The samples of the PCR seen here with green tips need to be mixed carefully. Once the sample has been mixed properly, using the pipettes here, 200 lambdas of the sample will be added to these containers. This contains enzymes such as lyase, which would destroy and lyse material inside the cell apart from genetic material. In this lab, the black machine is used more often, and that is because more samples can be processed by this device in lesser time. The advantage of using the white machine is that it can process samples individually. However, in the black machine, the whole set of samples need to be placed. So about 32 samples need to be placed in there. And it takes about 30 minutes for the black machine to yield results and about an hour for the white machine. We will now move on to RT-PCR itself. This is the kit currently used for SARS-CoV-2. The extraction kit, which we obtained from the previous step, will now be used. And 5 lambdas of this, which contains the extracted RNA of the virus, will be added to a master mix of 20 lambdas. The master mix contains enzymes and other substances. Enzymes such as Taq polymerase and reverse transcriptase, which are obviously required for the RT-PCR process. In this screen, we can see the results of the PCR samples here in real time. If the genes of the virus, such as the N gene and the E gene, are present, then the graph shows that the concentration of those genes have been peaked. The concentration of the genes is determined by the fluorescence that is detected by the device here. So this sample that we're looking at now shows a positive COVID-19 result.